Hello student, today I am going to discuss some general questions for general study related to biotechnology and science. So, first of all, science is fact, science is truth and science is knowledge. So, the first question related to general study, that is, what are the different products of fermentation? We can get acid also, gases also, alcohol also. So, answer will be they are different product, that is multiple product we are getting from fermentation. Next question, that is, who has discovered that yeast are responsible for fermentation? Louis Pasteur, a great scientist in the field of microbiology, had discovered the yeast are responsible for fermentation with the help of microscope and polarimeter. He has confirmed that yeast are responsible for fermentation. Next question is, who provided the chemical basis for the nature of alcoholic fermentation by using analytical technique for the estimation of carbons? Then you can say scientist was Lavoisier. Next question, that is, which of the following bacteria is not used for fermentation? You know, Lactobacillus, it is responsible for lactic acid, and Leuconostoc, Enterococcus. These are responsible for different product of fermentation, but E. coli is not directly involved for the fermentation. Next question, which of the following is the correct sequence or step for setting the curd? Curd is very useful. It is used as a probiotics. So you can produce the curd from the warm milk and put a small amount of curd as a bacteria that will initiate the fermentation after incubation. So correct answer is B. Why warm milk is used for curd preparation? Warm milk help the growth of bacteria. Lukewarm. Hot milk may kill the bacteria. Lukewarm milk, not hot milk. Very cold milk very render bacterial inactive. Means bacteria will not active at very low temperatures. So that very cold milk may render bacteria inactive. So all the reasons given above are true. Means if you want to produce a desired quality of milk, so you have to take refrigerated milk, then you have to warm at 100 degree, then cool at 37 degrees Celsius. Next questions, that is, which of the following not a product of fermentation? You can see idli, a dough of idli that require the fermentation, and dough of dosa that require fermentation, and bhature. Maybe the papad that require the dough not require the fermentation. Which what is the meaning of word extremophile? A bacteria that can grow at extreme environmental conditions, that is known as extremophile. So next question, that is, microbial fermentation is an example of bioprocessing. Bioprocessing means less useful compound that will produce more useful compound with the help of bacteria, that is known as bioprocessing. We can use the raw material as a straw or any juice of the fruits for producing the better quality of the product. Bioprocessing technique is the use of unicellular organism, yeast, enzyme, so that all the things are required during bioprocessing technology. Which are the following are the product of bioprocessing technology? You can get bread, pickle, wine, beer, human insulin, hepatitis B vaccine. So that we get all the type of product. So all of above is the correct answer. Next, what is cell culture technology? Means you are multiplying the cell in tissue culture media, growing the cell outside the living cells, living organisms. Means you are collecting the cell and put in the media, then we can produce the desired product. May have medicinal importance. What is cloning? Genetically identical molecules, genetically identical cell. This is also correct. Then genetically identical plants and animal. These are also correct. So the cloning means genetically and phenotypically. If they are similar, individuals are similar, microorganisms similar, then we can say it is cloning. SNCT stands for small nuclear cell transfer. This technique was used in making dolly. So generally, somatic cell, other cell of the nucleus of the other cell transfer in the egg cell apparatus, and then in animal cell culture media, there was a generation of dolly as a shape. SNCT involves, what do you mean by SNCT? Transfer of somatic nucleus to an enucleated ovum. So this is a very important technique for cloning. Why we are say cloning? Because we can we are utilizing the nucleus of the somatic cell, not the nucleus of the reproductive cell like ovum. We are removing the nucleus of the ovum and then we transfer the somatic cell, means udder cell nucleus of the shape into the cavity of or ovum structure. Then there was a animal cell culture and there was production of regeneration of the dolly. SNCT used to clone the animals like dolly. Okay. Next questions, that is RDT. RDT means recombinant DNA technology. Means you are going to utilize restriction enzyme, DNA ligase, just to cut the DNA and just to insert the foreign DNA and then ligate the DNA inside the plasmid. So plasmid is very important for the cloning of the gene inside the bacterial cells. RDT involves manipulation of DNA. You know, means you can change the gene, you can insert the gene, you can reduce the gene. Next, recombining the DNA from the different source. You can also join the different source of the DNA. Modification of plant and animal. Yes, you can also do. So all of above is the correct answer. Next question is, what is protein engineering? Modification of the protein using recombinant DNA technology. So if you want to change the structure and function of protein, so you have to change the gene of that particular protein. So if you change the codon of that particular gene, then only desirable change you can observe in the protein. In case of subtilisin, you can change the codon of methionine into alanine. Then we can get the desired quality of the subtilisin generally used in detergent industry. Better quality of subtilisin is index of best quality of detergent. What is biosensor? Biosensor means with the help of cell membrane and through the electrical subunits, we can monitor the amount of glucose, amount of any biochemical compounds. So biosensor has biological components. Yes, it is correct. Biosensor has transducer just to get the signals. Okay, then detect with high specificity means nanogram, picogram quantity we can detect with the help of biosensors. So all of above is the correct answers. What is nano biotechnology? Nano means 10 power minus 9 size of any molecules. So we can combine the nanotechnology means smaller size molecules with biomolecules. That is the correct answer to get a strong fibers, to get a bulletproof jackets in army. So these are the products of nanotechnology. Next, uh, we can say which of the following mammal cells are used to produce inactive polio virus? Correct. Chinese hamster ovary cells? No. Baby hamster kidney cell? No. Human lungs fibroblast? No. That is monkey kidney epithelial cell. So you have to remember such type of GK based questions related to biotechnology. We can be done to improve the crops. How can you improve the crops? By transferring the G resistant gene in cotton, BT cottons against insect cotton bollworm, disease resistant against any bacteria, fungus, virus, and abiotic resistance means that plant can grow in high salinity at high temperature and low temperature. So you can do all anything to make a transgenic crop, to make a resistant variety of the crops. So all of the above is the correct answer. Next question is which of the following is not synthesized in E. coli bacteria? You know insulin. We can insert the human gene inside the E. coli bacteria, inside the plasmid of E. coli bacteria, and we can multiply that gene and produce huge quantity of that protein, that is insulin protein. In the same way, we can also use the gene of human growth inside and put inside the plasmid of E. coli bacteria, and we can synthesize huge quantity of human growth hormones. Again, bovine growth hormones. We can also collect the gene from the animals that is responsible for bovine growth hormones and put insert inside the plasmid. That plasmid will become recombinant plasmid, and we can produce huge quantity through inserting that plasmid inside the bacterial cell, and by process of fermentation technology we can produce huge quantity of that products. So EPO, erythropoiesis, that is not going to synthesize by Escherichia coli. Next question, that is what is the main challenges of producing human protein in bacterial cells? If protein will be produced in bacteria, protein would have same structure as in human cells. Bacteria would turn into humans? No. All of above? No. So protein would have the same structure as in humans, just like human cell structure. The protein have the same similar structure like in bacterial cells. So we can easily produce that type of protein in huge quantity. That is that where the some question, that is GK based question, general study based questions. So you can utilize for any competitive exams. Thank you and have a nice day.